Hi, my name is James Bray, and I work in the Maiden Lab at the University of Oxford. In this talk, I'll be describing some of the latest developments regarding ribosomal MLST. RMLST is a universal bacterial sequence typing nomenclature system. Firstly, some background information. Multi-locus sequence typing is an approach to reduce the complexity of a genome sequence to an ordered set of numeric indexes in order to study bacterial populations. Ribosomal MLST indexes the DNA sequence variation of 53 protein-encoding ribosomal genes. Indexing with RMLST alleles allows bacterial genomes to be organized and analyzed efficiently with a particular focus on species annotation. As all bacteria have ribosomes, the RMLST typing system can be applied universally across the entire bacterial domain. Genome sequences and the DNA sequences of RMLST alleles are stored in a web-accessible database. We have compiled a large reference genome database. This database currently contains around 370,000 genomes from 12,000 different bacterial species. Genomes have been included from two different sources, the NCBI assembly database and genomes assembled in-house from sequence read archive data. All the genomes have been indexed with RMLST alleles, and there are currently over 2.8 million unique RMLST alleles across the 53 loci stored in the allele library. Taxonomic definitions are regularly updated from the NCBI taxonomy database. The species annotations of reference database entries have been validated, and incorrectly annotated entries are excluded. Here we have an isolate information page from the reference database to help visualize how RMLST works in practice. This page is for FAM18, a member of the Neisseria meningitata species. At the bottom of the page, highlighted with a red box, you can see the RMLST allele designations for this genome. Unique combinations of RMLST alleles are defined as ribosomal sequence types, assigned genus and species information, and saved in the RMLST profile library. This combination of alleles is defined as RST2380. There are now over 158,000 defined ribosomal sequence types in the library. We've created a species identification server, and this leverages the RMLST indexing of the thousands of bacterial genomes in the reference database. Here is a demonstration of the server using the genome of FAM18 from the previous slide. First, the genome sequence is uploaded. And the database of RMLST alleles is searched using BLASTn to find exact matches. When that's finished, the species of the genomes in the reference database that contain these RMLST alleles are extracted, and the lowest common taxon of each allele is calculated, and the final taxonomic results, along with the percentage of alleles that support the final decision, are reported. Here we can see the allele matches that were found by the server. I'll describe in a little more detail how this process works and how the lowest common taxon per allele match is calculated. Here we have a table that shows the species that were extracted from the reference database in the linked data values column. I've added a fourth column with the lowest common taxon for each allele match as highlighted in red. In the blue box on the left-hand side are some examples of how the calculation works. If there's only one species linked to the allelic match, this is the lowest common taxon. If there are two species in the same genus, then the result is that genus, for example Neisseria. If two species belong to different genera but share the same family, then the family node is the lowest common taxon, and so on. There is one rule exception, and that is to handle species annotations ending in SP. When combined with the species level annotation in the same genus, the species level annotation is the result. In this FAM18 example, 32 alleles support Neisseria meningitis and 23 support the genus node Neisseria. As Neisseria is an internal node within the taxonomic path to reach Neisseria meningitis, only the species level is reported, as highlighted in this red box. The allele support metric is defined as the percentage of all the matched alleles that have contributed directly to the reported taxa, in this case 32 out of 32, or 100% of the alleles support Neisseria meningitis. The MLST species identification server can be useful for detecting mixed samples. In this example, from an assembly from short-read data, the server finds alleles that support two different species, Neisseria meningitis and Arothia species. There are multiple allele matches to some of the MLST loci that correspond to these different species, highlighted here with the red boxes. This is a telltale sign of sample contamination. Extra evidence to support these findings is that the query genome size was over 4 megabases, which is much larger than the typical genome size for Neisseria meningitis. The MLST scheme has been implemented in some of the largest PubMLST isolate databases. This enables the species annotations of the submitted genomes to be verified, and also mixed or contaminated samples to be identified from the MLST allele designations. Some of these databases now contain tens of thousands of genome records. The species identification server can be run for genomes that are already in PubMLST using the MLST Species ID button that is found in the analysis toolbar at the bottom of every isolate page or from an isolate search, as highlighted here in red. The PubMLST website now makes it possible to perform phylogenetic analysis directly from a list of isolates that exist in the database. Here is a neighbor-joining phylogenetic tree of 70 Klebsiella and Raltella genomes across 17 species based on the concatenated DNA sequences from 51 MLST loci. Tree visualization is using interactive Tree of Life software, and the ITOL button is found on the analysis toolbar as highlighted here in red. This tree took 18 seconds to construct, as all the MLST allele designations had already been found and stored in the isolate database. The tree shows the isolate identifier, the isolate name, the species, and type strain information. Members of the same species cluster together within the tree. Six new species of Klebsiella have been defined in the last 4 years, and this analysis was part of a larger study to check the species annotations within the reference genome database. So to summarize, the MLST scheme can be used in multiple different ways to analyze bacterial genomes, identify species, and assess genome quality. I've described 3 different approaches, all linked through the common theme of defining and using MLST alleles. These are MLST sequence typing, phylogenetic tree analysis, and the MLST species identification server. I would like to thank Keith Jolly, Martin Maiden, and all my colleagues in the Maiden Lab, the PubMLST user community, and the Wellcome Trust for their continued financial support. and thank you for your attention.